This call is now being recorded.
Hello, can you hear me?
Yes, sir.
Yeah. You can see my slides also, no?
Yes, sir.
Yeah. Last class we were discussing about genetic engineering. Sorry, genetic code. Because genetic engineering is a biotechnology subject. Genetic code, and genetic code is a base for all those studies that you till 2020 that they have done. It's important part of molecular biology. So the genetic code, when we talked about the genetic code, I told many informations in the last class that is some vital characteristics of this genetic code and why this genetic code is so important for us. When we talk about genetic code, so first thing will come to our mind is triplet. It is a triplet, that means it's made up of three nucleotides. It codes for specific amino acid, and these codes are commaless. They are what we call, there is no punctuation mark, what we call commonly, and they are degenerating. All those issues are there. So today we know all those things, but there are people who work. That means what we can call it as a background workers. They are the one who deciphered genetic code. So deciphering genetic code was a big achievement in the field of molecular biology in the early of 1960s and 1970s. But later, today, if you look at molecular biology from the point of a genetic code, it's a very simple thing, and no need of spending too much. A simple instrument can give you all the data what you require to present as a genetic code, or maybe to see as a protein sequence. So it's very easy in the current scenario. It is very easy, but not during that time. So then you might be thinking, why do we have to study all those things? But when we study anything in science, we should know what was the background and how they discovered all those things. And this may not give you a perception of the present situation, what the molecular biologists are doing, but instead of that, what we will get is a kind of idea how people were working earlier. Can I pick up something like that, a very novel combination? Can I do something in the field of molecular biology or something like that? So that's why these old experiments are what we call classical experiments are always dealt in the class. That means it is a kind of a motivation or kind of a triggering to a new way of thinking molecular biology or something like that. Because we are all struggling a lot with molecular things today, and even the treatments of HIV or maybe the COVID-19 or maybe anything. So if you could target some of the gene or maybe some protein, so it is very easy to kill those organisms. So lots of work is happening in that field. And from a common man, if you ask what is the benefit of this, there is no direct benefit as such, but there is a lot of indirect benefits too. And today, what we study, most of you can see, to discover any drug, you have to identify what organism it is, what kind of infection it is, and where exactly it is, what are the symptoms, how much symptom, how extent it will go, then which molecule it binds. All those things we need. So that means a complete decoding is required at the molecular level to develop any drugs. So same thing. So that might be the way that people start thinking, and they might have thought that if you could develop a foundation from there, people can build up a knowledge in the field of molecular biology. And they never thought that it will reach up to a human genome project, what you see today, and we could, decode entire human genome. So they might have never thought like that, but the technology has helped us to lot. So then we will go to what is this deciphering genetic. Deciphering genetic code means the decoding genetic code. That's all. How they decoded a genetic code. Simply you cannot decode it, or you cannot say you need to prove chemically, or you need to prove biologically, or you need to prove experimentally that this is a triplet because this is the thing. So you need proof in biological science, right? So that's why deciphering of a genetic code was a great breakthrough there, and they were awarded Nobel Prize in the year of 1968, I think. 3-4 of them joined to be shared Nobel Prize for their great work. But today, if you see, it's a very simple thing, and people may laugh at that. So why they have done this simple work that time? But that time it was a great work and great achievement too. So now we will see the next what is this deciphering of the genetic code. See, when we go to a deciphering of the genetic code, we should understand first a concept of Ubel hypothesis. This Ubel hypothesis is nowhere related to deciphering a genetic code, but it's a part of it. That's what I wanted to tell. So, Ubel hypothesis, why it is important is, even though now when we talk about the three bases, we always tell two are important, the last base is not so important. So, this hypothesis is called Ubel hypothesis. So, it is put forward by Crick in 1996. So, it's a phenomenon in which a single RNA can recognize more than one codon, and this is due to the fact that the third base in the codon often fails to recognize specific complementary base in the anticodon. That means when you take from 5' to 3', if it is a 3 base pair, first two base pairs are important, the last base pair is not so important. So that means this is due to the fact that the third base in the codon often fail to recognize the specific complementary base in the anticodon. Ubling is attributed to the difference in the spatial arrangement of the 5' end of the anticodon. That means it's almost related to spatial arrangement, how the bases are organized. The possible pair of 5' end of base of anticodon, that is the tRNA, with 3' end base of codon is given anticodon. That means codon-anticodon base pairing is so important at the base 1 and base 2, and base 3 is not so important. That's what the Ubel says. Because if you look at a number of amino acids and number of possible triplet codon and compare with number of tRNA, tRNA are very less. tRNA are approximately 40, I feel. Yeah, maybe less than 45 or maybe there, not 60 and all. How come it is possible for one tRNA to do all these circles? It's because the third base pair is not true. So, Ubel hypothesis put forward by Crick. So, according to him, so single RNA can recognize more than one codon. That's what you call degeneracy, and this is due to the fact that the third base in the codon is not so important, and the first two bases are important. So, the pairing is happening between the first and second bases of tRNA with first and second bases of mRNA. So next, see, just please look at this picture, okay, carefully. You can see here, this is the tRNA for leucine. Leucine is one of the amino acids. It has got its own codon and anticodon. You can have a look here, mRNA from 5' to 3' end. You can see here, it's a sequence, the first picture, it's a sequence CUC. It's a normal pairing. GAG, anticodon, with the codon, it's a normal pairing. But it's same thing, if I look in the second one, there is no change in the anticodon, but there is a slight change in the CUU. Instead of CUC, it is CUU, but still it codes for leucine. That's what you call it degeneracy. That's why it's called Ubel pairing. Ubel pairing means the third base pair is not so important. First base pair is important, second base pair is important, the third one is not so important. So tRNA are identical, there is no change in tRNA. tRNA codes for leucine. So, but the change is only in the codon, that means in the mRNA. So, when there is a change in mRNA, maybe the third base pair, then nothing will happen. That's why a kind of a mutation in us might not change great extent if it maybe what you call morphologically, we cannot see much changes in us. Some mutations might not cause a major problem in us. So, it's because might be in the third one. If in third one there is a change, there's no problem. It codes for the same amino acid leucine and same amino acid as usual in the normal pairing. So if some mutation happened at the third base pair and it is C is replaced with U, no problem, because the final amino acid is leucine, so there is no change in the protein. If there is no change in the protein means there is no change in the action, it's a metabolic action. There is no change in the enzymatic action. There is no change in the overall biochemical pattern of a cell. So that pattern, there is no change, cell may not change or cell may not behave in a weird way. But if the same changes happen somewhere in the second or first, may create a problem here. Instead of leucine, it may cause for isoleucine. We don't know. If you look at the graph, you can find, the chart, you can find out what is the codon for isoleucine. So The base, that's why the Hubble hypothesis plays very significant when we study particularly translation, that's a protein synthesis, and the genetic code, decoding, all those things play a very significant role. So third base pair is not so important. So Hubble hypothesis explains the degeneracy of genetic code. That means existence of multiple codons for a single amino acid. So it's important. It's the one, if you look at this one, it's a characteristic of genetic code. Hubble hypothesis explains the degeneracy of genetic code, that is existence of multiple codons for a single amino acid. Although there are 61 codons per amino acid, the number of tRNA which they found out in the cell is less. It's due to Hubble or Hubble base pair. Either Hubble is responsible or Hubble base pair is responsible to have a less number of tRNA in case of 61 codons. That means less number of tRNA is sufficient to code for this, because third base pair is not so important, first two base pairs are important. So 40 tRNA can manage the entire process of synthesis. This might be what you call the benefits for the cell. So less tRNA production is needed. Otherwise, you need to synthesize more tRNA. That's also important, right? The more tRNA, if you put 20 more tRNA means extra energy, body has to send, extra nucleotide it has to absorb. All those things, it's well-connected network. So one is connected to another thing. If you chop up one, it will going to affect another thing. And there will affect the load on the human body also. The intake of nutrition maybe less is required. It's like how, if you look at the vehicle today, there was a BS4 vehicle earlier. Now the vehicle is BS6. Why it is BS6 means there is emission norms, Bharat standard. That means according to India, this should be the emission standard. This much should be carbon dioxide, carbon monoxide, sulfur dioxide, all those waste gases should be there. So that means you are optimizing the system. So this might be the part of optimization. That means by using less resource, get maximum results. That is benefit for the cell, that is benefit for the human beings, and that might have picked up during the course of evolution. So that's the important thing. So evolution play a very important role before we decide anything in biological system. So today what we say, ultimate organism, complete viable, what you call a highly intellectual organism like human being, or maybe animal like human beings. Today we might have got all this. It's because of changes or optimization of the system by our own environmental system, or maybe the course of evolutions. We changed a lot from old human being. If you look at a size of, but sometimes always it is a questionable. So if you look at Abu Simbel, that means Abu Simbel is a place in Egypt, I'm telling. Or else you look at a Karnak temple in Egypt, or maybe the Giza pyramid in Egypt, how precisely they were using around 3,000 years back, I feel. So they had intelligence. You cannot ignore the intelligence. That means we cannot compare always intelligence or human thinking capacity from the view of the modern human beings, what we call maybe the millennials or maybe 1,000 years back. There are people who are very active around 500 years back, 1,000 years back. People are very intelligent. They were using different technology, then they might have not had what technology we have today, maybe artificial intelligence or the mechanical support, but they had some other techniques. So that's why evolution is very important. So why I'm always stress on evolution is anything in biological system what we say, we need to connect with evolution. So this is all part of evolution. Antibiotic resistance in bacteria is part of evolution. It's a part of a survival strategy. Same thing might have developed in us. So Hubble hypothesis means first two base pairs are important, third one is not so important. So that's why less of the RNAs are there. And Hubble hypothesis explains the degeneracy of genetic code. Next, we are going to study the mainly deciphering genetic code, how they decipher. But here today I'm explaining two experiments. Are very simple, damn easy experiment, what they've done that time. But in that one, for exams, you study only one experiment, that is the corona's experiment. Corona. Argovin Corona's experiment. So the Indian born scientist experiment. But there is another person called Nirenberg and Matai. Their experiments are not need of studying. Because to understand the Corona's experiment, you need to understand Nirenberg's experiment. So that's why I'm giving importance to Nirenberg's too. So the cracking of genetic code began in 1961, the way back to 1961, with work from the American biochemist Marshall Nirenberg. So Marshall Nirenberg is a person from Greek, and they were discovered the structure, all those work were happening. Simultaneously, there is another person who thought of why can't I study the form of the protein. He started this. For the first time, Nirenberg and his colleague, that means I think Matai, were able to identify a specific nucleotide triplet that could correspond to particular amino acid. That's the important thing. A specific triplet, maybe UC codes for leucine. That means a specific nucleotide triplets that correspond to particular amino acid. That was their discovery. That's important thing. So for that, what they've done is, so they had a chemical synthetic technology. That time the people are very good in synthesizing something. So that is possible today also it is possible. You can synthesize polynucleotides or you can synthesize polypeptides in the lab. That means what work the cell is doing, that cell is taking one monomer and cell is taking another monomer, linking by a phosphodiester one. Same thing you can do it in a cell-free system. So cell-free system they have done. This Nirenberg and Corona, that means Hargobind Corona, Indian born scientist, both of them done. They work in a cell-free system. That's important. That's why in India the problem is we see things from the perspective of a biologist means biologist will only should study about biology. A chemist should always synthesize. So it's not like that. So it's end of the day your interest and your knowledge, your deep understanding of the concepts, phenomena, hypotheses, your interpretations, your bold guess, all those play a very significant role. Because many chemists they have done a great work in the field of biology. There are many biologists, they give a clue about chemistry, clue about physics, clue about many things. If you today see, if you see helicopters, commonly we give it, that is from a dragonfly. So dragonfly is the biological organism, right? It's insect. From there they got the idea. But there are many things. I mean, people used to say about bat. The people are working on the field, particularly the bat. Same technology, why can't we implement in the field of stealth fighters, fighter aircrafts? The people are thinking. So same changes are happening. People are thinking ultimately the end is something new should happen, something evolved, advanced things should happen in the field of biology as well as in the field of technology. Technology is helping us a lot to solve many issues today in today's case. But earlier they used to take years together. Now you can do it in a week together, maybe within a week. So first, what Nirenberg has done. So we need to concentrate a bit here. Nirenberg synthesized an mRNA molecule consisting only of nucleotide uracil. That means he prepared a chain of U. He prepared a chain U, and U. That means a poly U. So that mRNA is only U. So where did he prepare? He prepared a cell-free system. He found that polypeptide made up of these I mean, if it is a U, his prediction was UUU means phenylalanine. Only phenylalanine should be there. So he took UUU, connected the UUU, and made poly UUU. And you know, 3 UUU means it is phenylalanine. So he synthesized that in the cell-free system. From this experiment result made him to conclude that UUU might code for phenylalanine. So the thing is, you cannot exactly say that answer, but they told that this might code for that phenylalanine. Using the same approach, he was able to show that poly C. Earlier case, he took poly U, and he predicted then triplet U will code for phenylalanine. So he found out those the protein which is synthesized only of phenylalanine. Next, what he has done is he took poly C, so translated into polypeptide. It can exclusively consist of only proline. It consists of only one kind of amino acid, that is proline, suggesting that triplet CCC might code for proline. So first he synthesized poly U and found polypeptide only with phenylalanine. Next poly CC, and finally found out that it is code for proline. And he concluded if it is triplet is U, then it is phenylalanine. If it is CCC, proline. He just gave some kind of clue or some kind of idea. That's a very important thing what Nirenberg is that contributed to the field of what we call translation machinery or translation setup. So you can have a look here. Just I put one picture here. You can see there are poly U mRNA. If you translate that, you will get a polypeptide. Polypeptide is made up of only phenylalanine, 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 and phenylalanine. So from this he concluded, if U are there means it is phenylalanine. Another experiment he has done, that is poly CC. So he concluded that only proline. So this is the way how they build up that whole chain, whole 64 codon amino acid they build up by using this technology, simple technology. Poly U, poly C. Then this is a monomer which is made up of same nucleotide. If nucleotide is different, that work is done by Hargobind Khorana. Okay, Hargobind Khorana was an Indian-born scientist. He was working at Wisconsin. He extended the work of Nirenberg. He took the Nirenberg basic information and he extended that work with little more complex nucleotide. What he has done is, instead of UUU, he synthesized a polynucleotide with UC. He took U and he took C and added together. So he made alternating U, C. Earlier it was in Nirenberg, it UUU, C. So, but in case of Hargobind Khorana's experiment, the mRNA is UC. So when poly UC is translated in, he translated that. So he found instead of phenylalanine and proline, poly U code for phenylalanine, poly C code for proline. Here he found UCU, that means serine and leucine. He found serine and leucine here. Little strange, right? It's little strange because initial poly UU coded for phenylalanine, poly CCC code for what you call proline. But when you put it in the alternate format, UC, it is coding for serine and leucine. These and other results. So he has done other experiments also, similar experiment. So he confirmed that codon is triplet. That is the one thing he has done, a great work. That means he confirmed it. Yes, the codon means a triplet. And if codon is UCU, then it is serine. If codon is leucine, then it is CUC. UCU, CUC. UCU means serine, CUC means leucine. So that is the two things he could confirm by his simple experiment of polynucleotide consists of UCU, or UCUC, or UCU, That was his basic. So for this, I'll show you the picture. You can see here, U with C, U with C, U with C, like a it continues. So if you look at from one end to another end, if you read it properly, serine, leucine, serine, leucine. If you do it with different combinations, you can elucidate. Even he has done the same thing. He elucidated a complete strategy, the complete information which is needed to say UCU is serine and CUC is leucine. So this is the way how they deciphered a genetic code. And today we have a table of genetic code which represents what and all the amino acid. From these are the guys who has done a lot of work, who have done this work, and from them we could collect all the informations. And today everything is in the textbook format, and we could understand our basics. The primary basic concepts of molecular biology, molecular biology, or the translation setup in our cell system. So why this is important is DNA is found inside the nucleus. RNA destination is not clearly mentioned because it can be inside the nucleus or outside the nucleus, and protein synthesis is outside the nucleus. So it's very important. Information should decode from DNA, it has to convert into RNA, from RNA to amino acid specifically. So otherwise, if any changes that leads to a magnification change in the protein what it produces may have enormous effect on what we call in our physiological system or maybe in our biochemical system. So in order to avoid everything, so it is fixed, it is pre-planned, and it is clearly known to the cell system. So this is the way it has to go. And if this is the nucleotide, this is the amino acid. So that's why high-end precision is there. Today, in today's class, we started with Hubel hypothesis that's proposed by Francis Crick. So according to him, Ubel base pairing is important. That means anticodon codon base pairing. First two bases are not important. The third base pairing is so important. Sorry, first two bases are important. Third base pairing is not so important. So based on that, so the one, I mean there's a less number of tRNA compared to number of codons, and that also explains us the concept of degeneracy of genetic code. So degeneracy of genetic code means one codon can code for, or else one amino acid is coded by more than one codon. So one codon can code for one amino acid. But if one amino acid is coded by more codons, then we call it a phenomenon called as degeneracy. But if one codon is coding for one amino acid, we call it unambiguous nature of genetic. Then we studied deciphering of genetic code, that is the decoding of genetic code. There are two experiments we studied. One is a Nirenberg experiment, and second thing is a Hargovin-Coronas experiment. In Nirenberg experiment, what he has taken with the sulfate system, where he took poly U and poly C. He could find out phenylalanine, and then he could find out proline. U will code for phenylalanine, C will code for proline. But later, Hargovin-Corona modified little more, and I could say he extended the work of Nirenberg. He modified this. Instead of homogeneous chain, he took heterogeneous. That means instead of U, he took UC chain. So from there, he could identify UCU codes for serine, CUC codes for leucine. So somehow he gave clarity on part of, what you call genetic code. So both of them awarded, and three of them awarded a Nobel Prize in the year 1968, late 1960s, for their enormous contribution to the field of genetic. I hope you understood what is a genetic code, and